Yeah, so I'm going to give you a short introduction to the Genome Browser. I'll let you know what a Genome Browser is, tell you about the Genome Browser features, types of data you can view in a Genome Browser, and then end it with a short demo on show you how to access the VEuPathDB Genome Browser and how to get around in the Genome Browser. So why do we need a Genome Browser? If you download data or if you sequence data, what you get is a text file. So, and I've got here two examples. On the left-hand side is a FASTA file, which is just a DNA sequence. On the right-hand side is an ML file with the annotation, which you can, for instance, download from ENA. So extracting that information you're interested in from these text files is very time-consuming. You might also need coding skills. And for that reason, genome browsers have been developed. They make omics data accessible. You can view the data in a nice graphical format. And what you can see here is the genome browser that we are using called JBrowse. What you can see here on top is a menu. It tells you which genome is displayed. In this case, it's Toxoplasma gondii ME49, a genome that we will explore much better in the second exercise today. It also gives you a few options. You can change the genome, search for sequence, or upload your own track. Below the menu is a navigation area. First of all, it tells you exactly where you are on the chromosome with this rectangular red border thing. It also gives you an option to scroll along the genome, zoom in and out. With this option here, you can select different chromosomes or contigs, and you can search for genes or for areas on the chromosome. And below that is the genome and data track area. And the most important track is the annotated transcripts track. In red, we've got genomes that are going in the reverse direction, and in blue, in the forward direction. In white, we've got the UTRs, or pseudogenes are also in white, or non-coding RNAs. Omar also mentioned yesterday this select tracks button in his intro. If you select it, you get an extra menu where you can see how many tracks are mapped to this genome. In this case, ME49 has quite a high number, 762, but we have genomes with thousands. But then again, we have also genomes where just 10 tracks are mapped. So it's important then to use this contains text where you can search. Something we will explore in the first exercise. Okay, so what types of data can you find in a genome browser? One type of data I've already showed you is the annotated transcripts track, but you can also display the sequence. And this is a track called reference sequence. You need to zoom in to see the sequence exactly. And what you can see then is the amino acid sequence, the DNA sequence, and the forward strands and reverse strands. It's quite important sometimes to check the sequence itself. Maybe you notice that your gene, very often the methionine is not correctly predicted by automated prediction pipelines. And with this reference sequence track, you can then check if there is maybe another methionine nearby. What you can also view, and this is something that we are going to explore a lot today, are the transcript expression, RNA-seq data, where you can then also see it in blue and in red, depending on which direction. Another track which is useful is the synthetic sequences in genes, where the orthology is shaded then here. It's quite nice to just look at it and see if there are any discrepancies, if there is maybe a gene missing in one of the orthologs and in others. And in this case, I've chosen an area where you can see that ME49 is completely different than to all the other strains. You can see here this gene, this is ME49, has many exons, whereas the others only have one exon. In this case, it's even more that there are genes that go in the other direction, and ME49 has the one in the forward direction. And in this case, you just have to load more tracks and recheck which annotation is correct. And this is something that we are going to explore at length in the second exercise today. Variation data is also something that can be displayed. And this is something that we've covered yesterday in one of the exercises. I'm not going into more detail on that. and protein expression. So there is a track called where we pull together all the proteomics data sets. It's quite useful to see if your gene has protein expression. And just want to point out, we not only have genome browsers, but we also have protein browsers. And the main difference is that in genome browsers, you can look at the whole chromosome and genome, whereas in the protein browser, that focuses on just one protein. This is a view that you can access from the gene record page, and I'll show you in a minute. And it also shows you what kind of domains your gene has, your protein has, if it has transmembrane domains or signal peptides, and you can also select additional tracks, proteomic tracks. If you hover on it, you will see which interpro domain it is. And with that, I'm going to give you a short demo and show you how you can access the genome browser. So doing that also on ToxoDB, but it's the same on all of our sites. And you can just access the genome browser by the tools menu. And here's the genome browser. Another option on how to access it is from the gene record page. You might have noticed yesterday when you explored the gene record page that we have these buttons, view in JBrowse, and once you click on it, you get exactly the same view as you've got here. Already done that. And what you then get is this here, a JBrowse window. You can see here, this is the same view as shown here. I have not mentioned these two tracks, but we will cover them in the exercises. These are intron tracks, where you can see here if introns are matching the current annotation or not. And so this is something that we will explore at length in the second exercise. Also, this combined RNA-seq plot is something that we are going to explore. So just want to show you what you can do. You can, for instance, select additional chromosomes, other chromosomes, and once you do that, you can zoom in and out. You can also search, and this is something that we are going to do in the exercise. It's quite very slow because I've been given a quite big region. Okay, you can search for either you use the gene ID or you can just use product descriptions. So let's go and look for kinases. And you can see here, it gives you a list of all the kinases in this genome. And so click on it, you then get to that kinase. Here's this button that I've mentioned before. It's the Select Tracks button. It gives you an option to filter. Let's say I'm interested in the synteny track. I click on it. You just need to go back then to the browser and it will load the synteny track. Okay. And I think, yeah, and just want to point out here, you can change the chromosomes. Going back to the gene record page, not only here in that section, we've got the view in JBrowse genome browser, but in other sections too, you can, for instance, use the shortcuts. And here is the synteny track. And this little JBrowse window is in itself, you can also scroll along. You can also search for genes, but if you want to add additional tracks, you need to open it in JBrowse. Let's go to another shortcut, maybe the protein features. And this one here, it's a hypothetical protein, and it's true, it doesn't have any interpro domains. But you can open it then and view it as a protein browser. So that's all of the demo I want to give. And just go back here, tell you a bit about the exercise. So we are using TriTripDB. And this exercise, which is only 30 minutes, is only for you to learn the basics of JBrowse. You will learn how to navigate to a gene. how to add different tracks and explore them. We also have, in the very end, show you how to download data from JBrowse and how to upload your own tracks. And with that, if there are there any questions or anything that I've missed from other instructors that should be said?